Can I start the welcome, madam? Can I start the welcome address?
So please, sir.
Yes. Yes.
Sorry.
Good evening to everyone. I want to convey my sincere thanks and gratitude to our resource person, Professor Dr. Isabel, madam, for her commitment and hard work. First, we approached madam through my dear friend, classmate Dr. R. Govindarajan regarding our international virtual learning series on the first week of May. She kindly hearted Anburaj sir, கேக்குதா?
கேக்குது சார்.
She, madam kindly heartily accept our request and consent to deliver lecture for us. But unfortunately,
Yes.
she met an small
Yes.
accident, become immobilized for a few weeks. Today, madam is going to share her knowledge with us, even through she have some difficulties in the movement of her arms. Dear students, kindly note this commitment, sincerity, and hard work of madam. So even during her ill health, she want to fulfill her promise. Students, these kind of hard work and sincerity will take you to next level. Once again, I welcome, thank for accepting our request, madam. Very kind of you, madam.
Hello, I'm here just expecting the image of the presentation to come up.
No, madam, no.
Sure, madam.
You are
Okay,
sharing
now can you see the presentation?
Yes, yes, yes.
going on.
You can start the presentation, madam. Please, madam.
Okay, I will start.
Yeah, it is visible, madam. But it not from the first slide. Yeah, now it is okay. Just click on the first slide, madam.
I like to apologize with the group because there are some difficulties to make the projection. I don't know why today is not working properly. Well, I like to thank the organized committee to invite me for this webinar seminar, especially for the Arulmigu Kalahingam School of Pharmacy. Well, my name is Professor Isabel Campos Santaella. I'm from University of Orient, Venezuela. I like to talk about SARS-CoV-2, knowing the enemy. I'm trying to put this in presentation. Let me see. Can you see the presentation?
Yeah.
Yes, madam.
Well,
Madam, can you make it full?
it's not in full screen. I don't know why, but okay,
No, no,
I'll
it was
carry on. It's taking a while to change. Well, right now the world is facing one of the worst problems health-wise regarding this powerful virus. We know now a few things, but not enough because we haven't been able to find a cure or something that stop infecting our people. So in order to do that, we need to know the enemy. So now, right now, our enemy called SARS-CoV-2. But what is that? We need to know first what is a virus. Okay? What are viruses? Most people believe viruses are living organisms, but they are not. They are protein nature entities indeed, but they have the potential to infect other organisms. However, they don't have the ability of proper living organism to replicate. When an organism doesn't have that, we don't consider them like living organisms. However, it's a very clever form of life that can come alive when infect a host cell. Okay? Usually we can find different kind of viruses. They can be classified in many ways, but the problem is they don't have the power to replicate as I stated before. So how many kind of viruses can we find? Well, till now we have numerous kind of viruses, but thanks to the universe, only 1 to 2% of them are pathogenic to animals, plants, or human beings, fortunately. Well, because of that and their potential to hurt or contaminate other organisms, we think it is easier to fight against him. But it's not, because they are very specialized forms and got a highly ability to mutate and adapt to any sort of conditions. It can be physical condition, biological condition, and that gives a key, and maybe it's the reason they are still around because they've been for ages, thousands of years in the world. All right, then this genetic variability allow them to move forward and change according with the changes in the environment, and that makes very difficult to find something that can stop them growing or multiplying, even that they don't have the ability to do it by themselves, but they can still do it using another cell. Uh, okay. Let me see if I can move this down. Okay. Uh-huh. Finally. All right. Better. So now I like to show how we can classify them. We can do it using the features that characterize size, form, presence or absence of a particular cover or envelope, also the type of nucleic acid, and their ability to use a special host according with the susceptibility of that host. Okay? This shows, this picture can show the very different insight we can
Madam,
have. Oops,
sorry, madam.
sorry. Between
We can see the slide, madam.
bacteria, sorry,
It, it was stationary, madam. It not moved. Now you are showing the fourth slide too. Now we can able to see. You have moved now
okay,
only, madam.
sorry. All
Oh,
right.
thank you.
In this scheme, you can observe the size of a bacteria, which is 3,000 nanometers large, against these different forms of viruses. As you can see, there is a high variability in size between them. It can go from 20 to 300 nanometers. So, and that's one of the reasons there have been in history a delay in the study, because until this special microscope transmission microscope allow us to see 1,000 times bigger, we couldn't realize they were around us. Okay, and perhaps many diseases before were adjudicated to a different reason and were probably produced by a virus form. Their complexity goes further. You can see they can present icosahedral form, helicoidal, spherical, filamentous, and even have different forms that mix, like this bacteriophagous here, that looks like a small robot. This virus is able to infect other cells, like bacteria, okay? And if you see, it got a big head, which is icosahedral, and got a cylinder, like a body, large, and that is a helicoidal, it's a combination of both forms. They are quite specialized. If you see in this scheme, you can observe how the nucleic acid is inside the head in the kind of bacteriophagous complex form. And this one here is more similar to the virus we are talking about, is a SARS-like form. Okay? You can see the nucleic acid, you can see also the envelope and other internal parts of the cell. In this case, it's important to mention that for SARS-CoV-2, we got two kind of icosahedral viruses, the one that have no envelope and the one that have it, or the cover. Maybe you have heard the use of foam or alcohol liquids for cleaning hands and stuff. It's because these kind of detergents can dissolve completely this cover and put it, it's like you are no shield, you have no shield to defend yourself. All right. So, how many type of viruses we can get according with the host? There is a very, very long list. But I mentioned a few so you can relate the family of the virus and the host that can be invaded. For example, Cystoviridae is a family where main type of species attack bacteria. So happened with the Totiviridae family against protists and fungi, Pativiridae against plants and fungi, Rheoviridae, humans, animals, plants, insects, and so on. We can go through many families, but this is the one we are interested in, Coronaviridae. This is a family that usually was isolated from animals like bats and also some vertebrates, cold-blooded like snakes, for example, or lizards. But it's not usually common that one virus jump from one species to another. It takes a while to do that. I mean, years, many years. For us, we have a media time of life, 70, 80 years, it depends, okay? But now, for viruses, an hour can be many years, meaning they can reproduce very quickly when they infect a cell. And that's the reason we will see later on that when we get the virus in our body, we need to do any effort to get it out before it's able to install in our body completely and take over. The reason that happened has very close relation with the nucleic acid present. We can have viruses that got RNA and other got DNA. They also can have a single strand like this one, or can have double strand. That works for both. In the case of coronavirus or SARS-CoV-2, we will find a particular classification that, which is Baltimore classification, that show us taking all these criteria I just mentioned, like size, present or absent of envelope, the architecture of the genome, all these characteristics, and also if they have single strain, double strain, RNA, DNA, because it's very big, it's divided into slides. These are the DNA ones. We can find in DNA those that have icosahedral and complex form, like I mentioned, the bacteriophagous. They can be naked or enveloped. They can be classified according with that in Baltimore suggestion as groups. So we find group 1 till 4, as you can see in this slide. In the next slide, we are relating the RNA family. If we go here, with the same analysis, we can find RNA icosahedral form and helicoidal or helical, if you can say so. In the icosahedral, we find naked and envelope. In the helical, only those that got the envelope, okay? In this case, if we go through the envelope 1, which is the SARS-CoV-2, we will find him, or it, sorry, as group 4, okay? This, so mentalize now in this group characteristics. Just to make a kind of resume, I know all this information I'm giving to you probably is taking you to pull your hair because it's too much. I try to do it as simple as I could, but it's not as simple. So if we do a resume of that, we will have, according with the nucleic acid, DNA and RNA. They can be single-stranded or double-stranded. They can also be positive and negative. It has to be with a special charge that define like 5' and 3'. When the process of replication, inside the cell, it needs to be read that information, which is coded by gene, okay? I'm not going to give a biological class so long that you get mad, but it's important to understand which are the main stages that the virus has to go through in order to induce the infection. This gives us a very nice knowledge and which point we can attack. The best points to attack is the beginning when it's settling in the cell, okay, in the whole cell. But we can also attack inside the cell in different stages according with the map I will show you ahead. Okay. They are also, sorry, I didn't read this. The 5 main stages are the absorption is the stage when the virus attached to the receptor on the membrane of the whole cell. And then if they succeed, can fusion with the membrane and pass into the cell. Once it's inside, it will break the cover and release the nucleic acid with the information of the viral form. This can go different ways into the cells and use the enzymes and all the machinery, biological machinery for the replication, but in such a way that the host cells do not identify the virus as a different thing. So the immune system of the living organism don't attack it. That's a very clever way to invade a cell. But when the virus goes through the cell, use the enzymes, the energy for the process, and everything inside the cell to replicate, amplify the forms, and form the different parts of the virus. Then these parts, after being synthesized, have to assemble to form the pathogenic or the active form of the virus that can infect the nearby cells. So these particles called virions, okay? And these virions have to be released from the cell. How are they going to be released? They will be released by 2 ways. The lytic cycle or the lysogenic cycle. It means that they can break down and release the nucleic acid active, or they can get out of the cell in a vesicle and then release and attach to other cells. In this case of SARS-CoV-2, that's what it happens. They do it in a vesicle. I will show you in this diagram here how it happens. So just to remember, SARS-CoV-2 is an RNA virus, and this virus comes along attached. There is a glycosidation here. Through these spikes, the information of the virus, the infectious part is on those spikes. That's the reason always scientists make a lot of attention towards that. When they join together, this is the way, it's a perfect match. I mean, I don't know if you remember this toy we used when we were kids that have holes with different geometrical forms, and we need to put a cylinder, a cube, a star, okay? They fit one in another in a specific form. And that's the reason the host cell, which is this represent with I don't know what color you see it, it's like a lilac color. Then they release, after fusion with the membrane, release the strand. In this case, we are talking about RNA single-stranded positive, meaning it's 5' to 3'. Don't worry about that. This is biological stuff. But this happened. They got the basis that decode the information of the virus, and they form a template and copy, translate, and transcript all the information using the messengers and RNA forms for the replication procedure from the whole cell. When they amplify their information, this migrates to the endoplasmic reticulum, where there are the root part. When there are a lot of ribosomes, that's where it happens. When these copies are made, then they migrate to the Golgi apparatus and form the lipids. And these lipids are used to build the forms, the envelope, sorry, oops. Back to the pit. Okay. So after form the vesicles with the virion form inside, these vesicles have to be released. And this process called exocytosis. Well, the reason I present this, oh God, what happened? Oops. Mhm. Hello? 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 Are we back? Okay. Can you can you see the presentation? Okay. I'm back. I'm back. Something happened. Sorry, apologies. Okay. Uh, remember this part and some of this, and also this part, exocytosis, because many of the, let's say, vaccines and medicine that are being tried right now in trials concentrate or focus on this particular point, try to avoid as soon as possible the infection before the virus is built. If we stop the virus forming inside the cell before it assembles its parts, then we can reduce the amount of virus in blood and also reduce the symptoms, because there are little amount to produce the infection. We need a particular amount of what we call viral charge, okay, or some called index, and this is what produces the infection in people. So, just to clarify, some people use like synonymous, the name SARS-CoV-2 and COVID-19. Of course, if you say COVID-19, it's easier, but the real name of the virus is SARS-CoV-2. Why is that? Because the virus is which the etiological agent, the one that caused the infection, while COVID-19 is the name of the disease, okay? It's different things when we refer to it. At the beginning, we didn't know just that it was a coronavirus, but that we didn't know which one it was or anything about it. So it was called then nCoV 2019 because it was last year when it appears in Wuhan, China, as we will see next. It was identified in Wuhan about November, the end of November or the middle of November 2019. However, the World Health Organization just informed it on the 31st of December. So more than a half later, that is, it was a very, very big mistake. I'm not saying anything conspiration or anything about that. I don't go there. But if we have had the information before, we could have taken more care and stopped the spreading of the disease all over the world. In fact, too many flights and stuff from one country to another helped to spread that because nobody knew. Well, this virus is believed come from snakes and bats. However, as I mentioned before, a virus cannot just jump from one species and go to another and produce a contamination. Sometimes it takes 1,000 years to do that. Okay? In this case in particular, we believe there was an intermediary organism that helped to go through this quicker. And the suspect species is the pangolin. Okay? So this infection is highly contagious, and that's the reason it propagates quickly when we sneeze, when we cough. All these nanoparticles expelled during this process spread around, hit the clothes, the surface, any sort of format. It can be even our laptop. We are always on the phone, on the laptop, and if we sneeze and spread, somebody else can come along, use our computer, our phone, and get it in the hands. If you scratch an eye or take some food, we usually are working at the computer and eating something, a bite, and if you don't wash your hands properly, you can get into the body that form of contamination. The worst thing is that we can see the enemy. That's the biggest problem. If you can see the enemy, how can you fight it? And worse, you don't have any tools to fight it yet. If you go to war, we have a rifle, a pistol, or something to fight the enemy, but here we don't have that. We need to be more intelligent and so stop the spreading, uh, maintaining everything clean, being very careful with our hygiene and things like that, which are very simple things to do, but unfortunately not everybody follows. So how can we get infected? We see what I just mentioned in a way that is direct. We can call it direct contact when you touch somebody or something that is already contaminated. And that's the important thing. We do everything with our hands, and so our hands are always exposed to everything, especially in very young children and um, old people, that because of the age and stuff, move slower, don't take well care of themselves sometimes because have another kind of affections, health-wise talking, and also it can be when we touch something, as I mentioned, the laptop, the phone, or whatever, even food. So, and even when we just rock one of our pets, we do all the time. We got the dog, you know, we can acquire the virus these ways, very simple ways to do so. Till now, we have had detected this the presence of the virus in different ways. You will be amazed. Not only in saliva, we find it in all the system, mucus, nasopharynx, blood, tears, breast milk, semen, vaginal fluid, faces, hair, everywhere. That's the thing. In these pictures, you can see some samples of that breast milk. In fact, there are a few cases of neonates that have or recently born babies that have been fed with straight from the mother with breast milk, and they have got the COVID. Also, some people through the tears can get contaminated because this is the fluid, and then if you struck your eye, you can get it into, and that's a door to get in the body. It has been also detected in the bowels. These highlight forms you see here are viruses masses inside the gut. This is the intestine here. And well, it's been stained with the special technique, so you can see the highlight of the stuff. Well, this seems to be uh, very little things, but are very important things to consider because the virus is very clever and can survive for quite a while in different sources. It depends on humidity, temperature, pH. Uh, about of that, there are a few recommendations to try to have a alkaline diet, eating more fruit, or all this kind of fruit that is not only nutritional, is fresh food, and you can get the 100% of the antioxidants present on this sort of nutritious food. Well, the another thing is we can also use some or help ourselves using some solutions like alcohol, disinfectant for clothing. But we have to be careful with this because some people take things literally. Well, I don't want to mention Trump in this, but he made something in desperation, a quote. He said, "Can you just suggest to take something? I don't know, a drink of chlorine and get that thing out of our bodies." But some people hear that and start drinking, literally, and say, drinking those sort of things, isopropylic alcohol, chlorine solutions, and get intoxicated. The same happened with the use of chloroquine. They say chloroquine act to against the virus, and even people that didn't have the COVID-19 start taking it, and in a mad thought, they believe if I take two pills more, it will be quicker, and that's not the way it works. Medicine don't work like that, you know. They work by dose. If you take the right dose, it works. But if you take too much, you can get intoxicated or even dead. Or if you take less, it doesn't work because there is a right dose, the things to work. You know that because you are most of you are pharmaceutics or also chemists. Well, the main symptoms we can observe in this disease are, well, the common we see in most infections, and that was the reason at the beginning people thought it was just flu. No, didn't go further than that. So we can find people with fever, with cough, sore eyes, general discomfort, feeling bad. Some are vomiting or suffer, like a kind of fatigue. But the problem is, in 3 days, we have the chance to get rid of the virus before it is all causing the disease. We can do it as simple as make some gargarisms with salt or making the breath, holding the breath for 10 seconds. If you don't cough in that time, you are okay. You can do that every day and check yourself. Some people don't do it and say, no, it's just a flu. That's it. Afterwards, after 5 days, they start having some problems to breathe or lose the ability to smell things or the flavor, to feel the flavor. This called anosmia and dysgeusia, which is, the loss of smell and the loss of uh, taste. And then the acute respiratory syndrome start to settle and cause pneumonia and all the other things we have heard on the news. In fact, at the beginning, all efforts were concentrated on that. They say create the phlegma, attack the alveolus in the lungs, and uh, condensate water. The water blocked the absorption of oxygen and stuff. Later on, when happened the terrible Italian situation, they start forgot about the World Health Organization and start analyzing the corpse of the people that already died of COVID, and they realized there was phlebitis. Phlebitis is when the blood gets hard inside the vein and stop the fluid. But why this happened? This happened because the blood, hemoglobin, transports the oxygen to the lungs and everywhere in the body through the circulation process. However, if the um, iron that it contains in the center is taken out, that's what the virus, the SARS-CoV-2 does. It takes the iron in the hem molecule and use an inhibitor or function as inhibitor. So when the oxygen comes in the flux of the blood, it can fix or join to the iron because it's not there. This reduces the amount of oxygen in the blood. So by the time it reaches the different part of the body, the amount of oxygen reduced so much that the brain send an order to the bone marrow, please produce more hemoglobin because we need more. And this extreme amount of hemoglobin in the blood change density of the blood and produce clots. These clots reduce the circulation and start producing this phlebitis. And some people have had strokes. These we call thrombos. These thrombos can migrate through the circulating system and get to the heart, get to the brain, or produce a problem in the extremity, legs, arms. It depends where this happens, it can produce a stroke and also a respiratory failure major that produce a heart attack and the person can die. And that's what happened in the worst case. But all efforts at the beginning, at the time, well, nobody knew that that was learned at a very high cost a few months later. And well, right now, many people just using normal things for flu at the beginning of the stop the settling and nothing happened. But we will see there are few, um, this is just showing when it happens in the brain. And well, the rate of mortality at the beginning was said to be 0.1%. But now we know it's not like that. We already got over 500,000 people that dead because of this. In different conditions, the percentage is higher or is lower. So that is also being checking right now to see what is doing that some people resist better and others not. It seems that the natural condition, geographical position, and this kind of stuff uh, is better for some groups and for others no. It has too many variations. There are some I didn't mention here, but even in some patients have shown a change of the color of the skin from very pale to very dark because somehow in these particular cases the amount of melatonin has gone up so higher due to the COVID. So there is still too much to learn about this disease. It's actually a very, very complex situation we are confronting right now. If we see the distribution worldwide, well, this is the latest I could get. It's from April the 15th this year. If you see the distribution, the darkest color confirms the higher cases. Right now, well, after lower in some number of cases in most states in the United States, uh, right now, well, they flexibilize the rules and most people have contaminated. So you can see the darkest place or the darkest blue here is on United States right now, extending to Texas. By April, Italy was also in high trouble, and well, Russia and few other countries. So it's not just a very simple thing to fight on. So how can we detect this virus? Well, we just uh, can use biomolecular techniques to detect the presence of it. But we face some difficulties as well with that. First, when the virus is made out of RNA, the amount present is very, very little. So we can only detect the presence when there are a certain amount of it in the blood, okay? So, or in the saliva, it depends on the sample taken. And then what happens is that sometimes when you do the test, the person has been exposed for less than 24 hours. If that happens, or less than 3 days, the charge, the viral charge in the body is too low. So the sensitive of the test do not allow to register it as positive. But when you test the same person 5 days later, the person is positive. And then some try to say that is the test that is not good or whatever. So because of that, few countries have done some revisions about the results, and they have found out that sometimes it's because the test was using an old immunoglobulin in this case. For example, now that we are talking about India, we can see that in an experiment, 5 out of 13 cases show false positive for SARS-CoV-2 after use a very dated from 2017 immunoglobulin for dengue that was used as positive. This is the test is being used mostly as the first test is quick. In Singapore, 2 cases as well that suffered dengue were positive, but later on they were tested and they were really positive, but not for dengue. They were positive for COVID, and that was published recently on the Lancet journal of UK. So, well, there is piles of literature that can support how many variations this virus has shown, and for that, it's been very difficult to make assumptions. And well, everybody say it is Mr. Tedros from the WHO organization, who is guilty, but we can say that he knew less than he should know by that time. And as you know, political pressure sometimes take place, and it's not all in our hands, so better don't judge. Well, so let's go to a main important thing. So, what can we do? What drugs can we use? So, against SARS-CoV-2, if we use for the flu symptoms or common flu symptoms, aspirin, paracetamol, Theragrip, this kind of stuff, we can give comfort to the normal symptom as fever and as antipyretic. But a lot of experiments that have been carrying on right now have shown that, for example, nafamostat and cafamostat have action against in this particular point where the transferase is working against uh, the part of H2 of the spike of the virus. Chloroquine can act in 2 different points. It can act in the glycosylation at the point they join with the host cell, and this interferes with the angiotensin with binding. The angiotensin is a very important compound in the blood or in the cells, sorry, because some people suffer of high pressure and they take some medication that has to be acting against it, and this can interfere with that. And that might be the reason that at the beginning it were found some toxicity in some people. Hydroxychloroquine is less toxic, well, let's say toxic, but less powerful medication that is used for malaria, but it has shown that produce an endosomal acidification. If you remember that slide where I showed the viral join and different stages, I mentioned when it forms the vesicle, the exocytosis procedure. There are some steps there. We can see the effect of these drugs. The ivermectin is a common antibiotic used for parasites in pets, but it's very good antibiotic because it also stops the viral replication, which is good news. The same happened with remdesivir and favipiravir. These are 2 other antibiotics which show very good action. I will show you a slide, don't worry. Just want to introduce you to some terms that are in another slide. Diclofenac, you can have it sodic or potassium, different mixtures, acts against the inflammation that is produced in the lung with the coughing and the respiratory system. Dexamethasone also improves that, but it acts really stopping the release of substances that can produce inflammation. Some people have certain kind of diseases that are interferes with the immune system. That means that the own immune system attack their own body because did not recognize as they belong to the body. So, like people that suffer lupus or HIV and certain kind of these immune related or that can suppress or the treatment sometimes is the one that suppress the immune system and can produce another kind of all this stuff has to be taken in mind or bearing in mind. Imatinib is an anti-cancer drug that is also used uh, and it attacking a point to avoid the release of the cell. Exactly the point of the endocytosis I mentioned before. If we go to this slide, I can illustrate better what I mean. Here is the virus, the SARS-CoV-2, goes here. Chloroquine can act here, attacking the ACA2. Nafamostat and camostat can use here as well, but this time is on the part of the joint binding procedure when the the spike join by glycosylation to the receptor in the membrane of the host cell and divides or clydes into S2 and S1. Particularly, these two medicines act to the S2 part, okay, avoiding the attachment. So this is an important point, a very early moment to stop it. The second is when it is released and it's copied, remdesivir and rapipapil can act here in the replication complex, so avoiding the transcription. Cyclosporin A as well can do that, but it requires it's less effective. I mean, if we go through the whole way, we can also stop using this other compound like lopinavir, ritonavir. They act exactly in this point when the copy is being made and the triclonation pro, and disulfiram when it's also in PL4. So avoiding the proteolysis here. So if this change doesn't take place, it can continue the procedure for the formation of some of the part of the virus. So if the process continue, we can also affect the exocytosis procedure using imatinib, for example. So these are few examples of them. I think I'm taking more time than I should. Uh, I will try to go quicker. These are just some prevention measures we can use with the different type of masks, shields. Well, unfortunately, not all the masks work. If we check the size of the virus, it's so little they can go through most of the different masks already made. The best masks right now are the N95. Well, there are different types indeed for that, and the mixture of them, like you can see, this is an N95, this is an N95. Some people, because they ran out all over the world because of the pandemic, they start doing them handmade. I mean, not the official one that you can buy in the pharmacy. But these also stop the particles and virus can fix in dust, in fibers, whatever. And if we have the mask and somebody else cough over us, the mask protect us. Usually, just the person that is got the disease should use it, but it's better because of the highly contagious. virulence of this virus, it's better everybody use it, okay? And follow the biosecurity measures like washing hands, using alcohol substances to, if you don't have water and soap close to you, personal hygiene like a daily bath, cleaning tooth, blow your nose, and wash hands all time, very frequently during 30 seconds. If you keep your home clean as well, and everything you get into the house, you go buy food, you clean the bag, you clean the inside, you wash the vegetables, organize very well the clothes you are wearing, you take it off, and have a shower up before you continue anything. So you avoid, especially if you have kids or old people in your home, and then you avoid the virus to get into your home. Okay. Well, there are many things we could do apart from the hygiene. Avoid direct contact with people. Also, we should, um, if we feel any breath difficulties, and if you take any anti-allergic medicine and it doesn't work, you should go straight, especially if you've got temperature and this kind of symptoms. So it's better prevent those things and allow medical doctors and nurses with know the procedure, the right procedure to do to help you out. I know it's a scary time, and sometimes you could feel, no, I don't want to go to the hospital. I will die there. Remember, get the infection doesn't mean you're going to die. Okay? It could happen, but you have to take well care of yourself. Be responsible. Follow the distancing, follow the quarantine, and whatever the health authorities suggest to do, because they are the best people to know how is the right thing to do. As final remarks, I like to suggest that controlling disease like this is very difficult. But we have many things in favor of us. We got technology, we got people that have the knowledge to do so, we have resources to do so. There are few countries like ours in Venezuela right now. We are rich, but the money is in somebody else's pocket, unfortunately. And well, we need to do the best we have. But don't, my message is personal is not to be afraid. Do the best you can do. Follow rules. I know sometimes you feel like no, especially in India, you got a very high population there, and you know you are facing a very high mother nightmare right now, but everything is gonna be fine. Human beings have always defied everything, but we need from here to take responsibility for our actions. We need to care about the environment. If the environment is balanced, all these species will also be and won't attack us. If we don't care about environment, the environment will take care of us. That's my thought. So, well, this is the presentation I have for you. I know there is a lot still to say, and you must have a lot of questions. Forgive me if something was not very clear to you because I am a biologist as well, and I am used to this stuff. So perhaps I avoid to say some bits, and well, we can go there. I like to thank you all, and well, we can get some questions from you, and I'll do my best to answer all of them. I hope.
Thank
Do
you,
you have?
madam.
Okay.
Really, we had a wonderful and informative, nice presentation, madam.
Well, you're welcome.
Thank you, madam.
Do you have any questions?
Hello.
Do you have any questions?
Any questions, students?
Good evening, madam.
Good evening.
If we improve our immunity, we can able to avoid that COVID, madam.
Sorry, could you repeat the question, please?
Yeah, amazing.
You can type the question if you prefer.
Yes, madam.
Hello, ma'am. Ma'am,
Hello.
good evening, ma'am. Good evening, ma'am.
Good evening.
Ma'am, this is Arjun from AKCP, ma'am. Yeah, ma'am. Actually, I have one
Okay.
question, ma'am.
How old?
Ma'am, actually, WHO declared that COVID-19 is going to be an airborne disease by yesterday. Is it so, ma'am?
Sorry, could you type the question? I can't understand well. There is a lot of noise down there.
Ma'am, actually, yesterday WHO declared COVID-19 is a airborne disease. So we have to use our mask for all the times. So we are supposed to use the mask for all the times.
Yes, you
What are
should
the
use the mask all the time. When
yeah
you are out of your home,
yeah yes
inside
ma'am
your home, you can be free.
yeah sure
Okay?
ma'am. Whenever we are going to outside the home, we have to use the mask. So what is my question is, so what are the side effects by using a mask for all the time? Is there any side effects for using a mask for all the time?
Oh,
If yes,
side
what are the
effects.
side
No.
effects?
Well, there have been a lot of opinions about that. Some people said that they can breathe with the mask or they can feel fatigue or whatever, but there is nothing proof about that. Okay? Of course,
Okay,
if
ma'am.
you are in a place where there is a lot of heat, you will feel that you cannot breathe because this is quite tight, and if you got 30 degrees, 40 degrees temperature outside and you are
Yes,
walking or moving or doing effort, you will feel like lack of breath, isn't it?
yes, ma'am.
But it's not related exactly with the mask. It's a consequence, of course. And for medical people that have to wear all these masks and shield all day long, they even have skin problems because their skin can't breathe, you know,
Yes,
because
ma'am.
they pass too many hours wearing that. So they are having problems. But the common people just complain about some stuff that is not really related with the mask or with the
Okay.
use of the mask.
Thank you, ma'am. Ma'am, I have one more question, ma'am.
Yes.
Actually, you have mentioned that the virus is going to survive in an inanimate object for few hours. So how
You will
long it
speak
will
very.
survive? Yeah, you have mentioned in your slide, ma'am. Actually, my question is, how long it is going to survive in inanimate object, ma'am? Like hard surface or any soft surface. How long it is going to be survive,
How
ma'am?
long?
Yeah, yes, ma'am.
Well, it depends. In clothing, it can last 8 hours, 9 hours. It depends of the material it's made the clothes. As thicker it stay longer. You see?
Okay, ma'am. Yes, ma'am.
If it's the surface, it can last depending on the humidity of the environment. If it's
Okay,
very
ma'am.
humid, it will last 10, 12 hours. You see?
Yes, ma'am. Actually, ma'am, in US, there is a increased number of cases because of its uh, low relative humidity in US. In
Yes.
India, we are having summer, ma'am. Now we are in summer, around 40 to 45 degrees Celsius in our country. In summer
Well,
cases
uh,
also, we are increasing the infected persons ratio. So daily basis, we are having lot of patients, even though we are in summer condition. So why, ma'am?
well, the reason is you got too many people outside. The distance is important. So the virus is already everywhere.
Okay, ma'am.
Do you understand? The
Yes.
virus right now is already everywhere. So you cannot see it. So you
Yeah.
can get it anywhere. You touch a table, you hold something, somebody touch you, somebody push you, you sit in a seat where somebody else contaminated sat before, you can get the virus. So the important thing is when you get home, you get clean immediately. So you don't let it settle on you. But a lot of people is not being acting responsibly. You know,
Okay.
they are going to the beach, they're doing parties, they are drinking alcohol. This time, we should take it as a very high proof for us not taking anything that can harm or diminish our immune system.
Okay, ma'am. Thank you, ma'am.
Okay? You're
Madam,
welcome.
there is a question in the chat box. Why people, why that virus is affecting only human beings? Why the virus is affecting human beings? How the animal has been left out?
Okay, I can understand well the question. Some people is leaving the reunion. Okay. Could you type the question, please?
That,
Hello.
that virus is affecting human beings only, madam.
How the virus affect human
No, no, no,
beings?
no, no, no. COVID is affecting human beings only. Why it is not affecting
No,
any animals?
no. Now I got your question now. No, right now it's affecting also pets like dogs, cats, and we have to be careful with them as well. So you cannot um, give a bath to a pet every day because you want to kill the pet. But you can use deodorizing substances. You can avoid other people touching your pet. You see, you can, for example, I got a dog, a big dog actually, and it's a golden retriever. So when my husband goes to walk with him, he doesn't talk to anybody or say hello far distance. Don't allow anybody to touch our dog. When they came in home, my husband spread a little foam solution in his feet, clean with a tool, a cloth, and then they get in. My husband take his clothes, discharge in a bag, and go and have a shower. Okay?
In COVID-19, the exposure time has the effect, madam.
Well, the disposal time, well, the viral charge is very important. For example, if you touch a surface that is contaminated, you can, if you don't touch eyes, mouth, or nose, you can keep it away using just a simple solution with alcohol, a gel, one of these hand gels you can get everywhere. If there is no water, because that's another fact, many populations don't have water. or availability of water at home, like have to walk a very long distance to get that water. So you can clear yourself using one of these health solutions, uh, but most of them got alcohol in it. If you use too much, because some places, if you go to a supermarket, they got gel for you before you get in the supermarket. And then if you go to another one and another one, you are getting a lot of that gel in your hands. So that is not good as well. So as soon as you can, you have to wash your hands with soap and water, and avoid touching face or anybody as much as we can. That's the reason the reunion with many people or go to church, to class, and everything has been stopped to avoid, because how you can tell a little kid, don't touch that, don't touch that. They touch everything. Kids touch just everything. The roof, the table, the hair, the face, somebody else's. You can avoid or tell a little child to stop being themselves. It's going to be a very big challenge, actually, to overcome this pandemic, I believe. And I regret to say that many people, after the main top reach us, will die, because when you have a respiratory tube inside for so many days, you lost a lot of weight because you cannot eat. Your food is intravenous. You lost 40% of your weight. You lost 20% of your ability to breathe properly. So all of these will be negative for your future longevity.
Thank you, madam.
Okay. Any other question?
Thank you, madam. Very. Hello. Thank you, madam.
Hello?
Thank you, madam.
Well, thank you to you all, especially
Madam.
you and Mark and Narayanam.
Madam.
It's been a pleasure for me to hold this conference for you.
Isabel, Dr. Isabel, madam. You may be busy
Sorry?
with your madam. Can you
Yes?
hear?
Yes, sir.
Well, I can hear a few voices, so I don't understand very well.
So you may be busy with your work, madam. Must you look after your health, madam. Take care of your health. Thank you so much for your kind support for the development of our students' career, madam. In this occasion, we are expecting your cooperation and support in future endeavors, madam. Thanking you, madam.
Okay, well,
Thank you.
very thank you to you as well.
Thank you, madam. Take
Bye
care,
bye.
madam. Take care. Take care. Good
You
day.
too. Bye bye.
Thank you, madam. Can we close the session, madam? Thank you, madam. Bye.